So welcome to all participants. Welcome from the International Uveitis Study Group. Since January, we have webinars regularly, always the first Saturday of the month. But now, due to the topic, I think we decided to have an additional webinar. It's about COVID-19 and the intraocular inflammations. Well, there's hardly any other topic we are talking about since years. And I think it's very, very important to be aware of COVID-19 as ophthalmologists and to work together with other specialties. Even now that we have vaccination against COVID, we still have new problems. And that was the reason why we really decided we should have an extra webinar for COVID-19. Well, I have to thank all the participants. I wish you very good information. I think you will get that. I have to tell you that we do not have any CME points for this meeting here, webinar, because the time was definitely too short. So you will not receive, as regularly, any evaluation sheet to send back. Enjoy your meeting. Vishali, you take over.
Thank you. Thank you, Manfred, and welcome all the participants to this special webinar on COVID. We look forward to lots of questions which our esteemed panel would be very happy to discuss, both individually to you as well as in the panel. With this, I'm totally privileged to invite Yolanda, who is the best person to talk about virology of SARS-CoV-2, because understanding the virology is the most important aspect of understanding the disease that it produces in the eye. Over to you, Yolanda.
Thank you, Vishali, for the nice introduction. Thank you also, Manfred, for the invitation. I will share my screen now. So,
yes,
can you see this? Great. Okay, my name is Yolanda de Groot-Meijnes, and I'm a molecular microbiologist at the University Medical Center in Utrecht, the Netherlands. And I always had a special interest in uveitis. I'd like to acknowledge my husband, Raoul de Groot, who provided me with some of the slides. He is almost 35-year practicing coronavirologist, so I'm close to the source, so to speak. SARS-CoV-2 is a member of the Coronaviridae, and the Coronaviridae represents viruses of mammals and birds, and it consists of, this family consists of 4 genera, 25 subgenera, and 43 officially classified species. And SARS-CoV-2 is a member of the beta coronavirus subgenus, represented here, and a member of the sarbecovirus subgenus, represented here. And on the right red dot is SARS-CoV, the first one, and on the left is SARS-CoV-2, the current circulating virus. Other familiar human coronaviruses are the more common cold viruses such as 229E, OC43, HKU1, and NL63. But these are represented in other genuses and genera. Now, SARS-CoV-2 and the COVID-19 pandemic has been going on for almost 2 years now, and I looked up the numbers yesterday, and we now have almost 240 million confirmed cases, and almost 5 million deaths. Now, SARS-CoV-2 is an enveloped positive strand RNA virus, and on the right you can see a schematic representation of the virus. It consists of a plus strand single-stranded RNA, which is protected by the nucleocapsid protein in red. Then this is enveloped by a lipid bilayer, so from the same origin as membranes inside the cells. And within this lipid bilayer, the M protein is embedded, which is involved in creating the structure of the virus, the minor protein E, and the notorious spike protein, the yellow one here. And the spike protein is also visible in EM pictures shown here, and that causes in EM a kind of corona around the body of the virus, and that's where the family got its name from, coronaviruses, so not from the beer. Then the Genome Organization is represented schematically here. The genome is approximately 30 kilobases in length, which is very large for RNA viruses, and the coronaviruses are among the largest of the RNA viruses. It is for 2/3 covered by an open reading frame 1AB, which harbors genes for proteins involved in replication and transcription translation. And then 1/3 is reserved for other open reading frames, which express the structural proteins, the spike, the E, the M, and the N. But it also the genome also harbors genes for accessory function, and these generally counteract immediate host responses. And these genes are expressed from subgenomic mRNAs depicted here. So on the subgenomic RNA, the first open reading frame is translated and not the ones downstream. Now, the virus life cycle starts with attachment to the plasma membrane of the host cell, and crucial for this is an appropriate receptor. The spike protein binds to, in this case, ACE2, and another cell host protease, Tempus 2, is also required, and I'll come to that later. Upon binding and protease function, the lipid envelope of the virus will fuse with either the plasma membrane or the membrane of the endosome. And then the viral genome is released into the cytoplasm of the cell. And from there, replication of the genome will take place in specialized compartments. These are made by the virus through rearranging the architecture of the cell. And from offspring genomic RNA, the subgenomic RNAs are produced. These then are used for translation of the various proteins. The structural proteins come together with the newly made genomic plus strand genomic RNA, and here the viral particles are produced, and these are then released in the extracellular environment. Now, I would like to tell you more about SARS-CoV-2 entry in host cells that depends on the presence, the surface expression of ACE2 and Tempus 2. And ACE2 is an angiotensin converting enzyme 2, and it's a very important protein in vascular homeostasis. Now, this is a schematic representation of the Corona SARS-CoV-2 spike protein. It consists of various domains, most notably domain B, also known as the receptor binding domain, on which many vaccines are based. And this little yellow part, which is the fusion peptide. Now here you can see cryo-EM depictions of the spike protein. The spike protein is a trimer, and each of the monomers are color indicated here. And it can, each monomer can adapt two isoforms. The one with the RBD domain is this B domain in the down position, which is depicted here, and the RBD in the up position. And you can see here this part sticking out. This is the RBD. And also you can see it here. This is the RBD in the down position. This is the RBD in the up position. Now, the RBD in the up position is an unstable form, but occurs every now and then. And this is a prerequisite for the spike protein to be able to bind to ACE2. So in this condition, the protein will not bind to ACE2. In this condition, it will. So the steps in coronavirus infection are binding of the spike RBD to ACE2, which is shown here. And then the cell surface protease, Tempus 2, will cleave at this position. And this will release the fusion peptide. And this fusion peptide, which is hydrophobic, will then insert into the plasma membrane. And this will cause the envelope of the virus to fuse with the plasma membrane. And then the genome can be released in the cytoplasm, and the replication cycle can start. So these are key events to take place. Otherwise, the virus will not be able to infect a host cell. Now, how are ACE2 and Tempus 2 distributed in the body? Well, we know that the virus can infect the respiratory tract, and indeed both proteins are expressed in the upper respiratory tract and the lower respiratory tract, but also both proteins have been found to be expressed in the brain, in the heart, in the liver, in the kidney, and in the intestines. So theoretically, the virus could also replicate in those areas if it can reach those areas. Now, this has also been looked at in the eye, and co-expression of both proteins has been found in the conjunctiva and the limbus, the cornea and the retina. But I'm not going to go into detail because I think the other speakers will discuss this. Now, another hot topic on SARS-CoV-2 are the variants. With high replication rates of a virus, variants can occur, in particular among coronavirus, because they are notoriously they're notorious for mutating. They have a high mutation rate. And compared to the original virus, we already have had several variants taking over, also called variants of concern. And most notably, these are the alpha variant, the beta variant, the gamma variant, and now the delta variant. And these variants have mutations in all over the genome, but the ones of most interest are the ones located in the spike protein, and then particularly in the receptor binding domain. And this is an overview of key mutations in these 4 or 5 variants, and they are all related to a higher affinity to ACE2. And this has clinical impacts on the transmissibility of the virus, which goes up. It can have a higher pathogenicity, and there is a risk of immune escape because a lot of vaccines are spike-based or even receptor binding domain-based. Well, as we all know, the virus is transmitted through respiratory excrements, and also it is also found in the feces. Although we think that the oral fecal route is not of very large importance. Probably the most important route of transmission is through respiratory droplets, which can be transmitted through direct contact or at surfaces. But more importantly, the droplets can be transmitted when people are close together. Hence all the restrictions, the distancing advices. But another way is through aerosols. Aerosols are a lot smaller than the respiratory droplets, and these are airborne, so they can travel over a longer distance. And particularly in poorly ventilated areas where people remain for prolonged periods of times, the aerosols can be a very important route of transmission. Then the disease. So SARS-CoV-2 is the virus, and this causes coronavirus disease, or COVID-19. Most manifestations are respiratory. The virus can cause upper respiratory tract symptoms like rhinitis, anosmia. It's a very important clinical feature, rhinorrhea, sore throat, and in the lower respiratory tract, it can cause, particularly in severe cases, pneumonia or even ARDS, and can cause death. There are also atypical systemic symptoms like malaise, fever, but also outside the respiratory tract, the virus has manifestations. It can cause heart arrhythmia. It can also cause neurological symptoms like impaired consciousness, but also muscle pain, problems in the digestive tract. Particularly the neurological symptoms like impaired consciousness, lower cognitive performance, and arrhythmia are also symptoms related to what we call long COVID, so they can manifest over longer periods of time. Then what is the underlying mechanism of more severe coronavirus disease? Well, in the lower respiratory tract, the virus will infect particularly type 2 pneumocytes. These cells have express a lot of ACE2 and Tempus 2. This will cause an activation of the immune system, and this may be that severe that a cytokine storm will occur, leading to eventually even leading to ARDS, so acute respiratory distress syndrome, this may even cause death, as mentioned before. So this starts with the infection of the pneumocytes. But also outside the lung, COVID disease may manifest, and the other that is initiated by infection of endothelial cells. This can cause endotheliitis and has been associated with a downregulation of ACE2, and this can cause a coagulation imbalance, causing coagulopathy and vasculopathy. This by the production of thrombi, which can cause embolisms throughout the body. And this may also play a role in ocular disease. So the key points I'd like to stress is that the spike protein is key protein for virus entry. Host cell entry depends on surface expression of ACE2 and Tempus 2. And SARS-CoV-2 can cause severe respiratory disease, but also systemically coagulopathy and vasculopathy. I thank you for your attention.
Thank you very much, Yolanda, for making this very complex virus very clear to us. Thank you. There is a question for you in Q&A that you can answer. And with this, I move on to our next speaker, Dr. Rupesh Agarwal from Singapore, who shall be talking about, can SARS-CoV-2 be detected in the eye? Over to you, Rupesh.
Thank you, Dr. Vishali. Am I audible? Slides are okay?
Absolutely.
Good. Thanks. I think Yolanda has set the stage and spoken about the virus. Now, I don't know whether I can really answer this, can SARS-CoV-2 be detected in the eye, but it's rather a controversial question. And I will try to put in some of the points which we have learned over last 1.5 years through some of the work which we did and some of the work some of our colleagues did. I think just drawing your attention to what Yolanda spoke about, can the coronavirus disease be affected and can it? So we did wrote about this article when on January 23rd, the first case, 2020, the first case came in Singapore. And when I reached out to my colleagues in infectious disease department, it was the same question, can it affect the eye? And you can see the interest, and this paper has been cited 523 times, and we did original research about looking at the tears and has been cited 360 times in last 18 months. You can imagine the interest which people are having about this particular topic. And let's try to understand how what we have learned in this last 1.5 years about this virus. So again, the typical questions are, what are the ocular manifestations, whether it is ocular surface or intraocular? Does it affect the eye, inside the eye, in the terms of retina, choroid? Can it transmit through the tears? And can it, the conjunctival secretions, secrete SARS-CoV-2 virus? And if so, what are the proposed mechanism? So let's try to kind of dissect it into different segments. So A, can coronavirus disease affect the surface of the eye? And what are the ocular surface manifestations? In this systematic review, which we published sometime before, we did mention about all the possible thing which can be there with the coronavirus. And this is again not exhaustive. There are more reports which are coming out, whether it is a conjunctivitis, photophobia, blurring of the vision, itching, dry eyes, the foreign body sensation. And we did reported about even the conjunctivitis which we saw in a patients of coronavirus. Besides that, the intraocular manifestations or some of the other ocular manifestations can be refractive errors, which we have also reported. And we have seen that people, the patients with COVID can get refractive error. They can get intraocular inflammation, which Dr. Vishali will be speaking in detail later on. But in addition to that, the group have reported on retinal microangiopathy, retinal vascular occlusion, choroidal vascular changes, and optic neuritis. And these are some of the diagrammatic representation about the refractive error changes and the loss of accommodation as presented in this illustration, or the retinal microvascular changes. So we got a fair bit of idea that there is something happening in the eye. Now, why it is happening, what it's making happen, let's try to go it a bit deeper. The next question comes to us is, can SARS-CoV-2 be transmitted via tears and conjunctival secretion? Again, my dear friend Andrea from Italy will be speaking in detail in the next talk. But again, just to bring it up, why do we need to know that? Is to get us whether the PPE or some of the infection control protocols are required in the eye clinic, whether the outpatient procedures to be carried out, particularly like Goldman applanation tonometry, or can tear sampling be used as an adjunct to diagnose coronavirus, not only in the eye, but also in the body, and also for the treatment. So you can imagine if we can diagnose or if we can treat coronavirus by looking at the eye, I think it can help us quite significantly. Now, if at all it happens, what are the possible theories for a virus to be manifesting in the conjunctival secretions? A, it can be either because of direct inoculation from the infection or from the virus itself into the droplets, or B, it can be migration from the respiratory tract onto the through the nasolacrimal duct onto the conjunctival surface, or C, it can be the hematogenous infection through the lacrimal gland, and D, transmission of the tears to the respiratory tract by drainage through the nasolacrimal ducts. So there are all possible mechanisms by virtue of which you can see the virus on the surface of the eye. And again, this has not been detailed out in many studies, but we'll hear from Andrea later on in his talk. There are different things by techniques by which you can detect viral RNA. And again, in that, I like to classify into two things. One is the method of sampling, and there are people who have been doing direct conjunctival sampling or conjunctival swabs. Our group have done Sars test strips, but again, this itself becomes a topic by itself. The comparison
Rupesh,
between the
video is kind of.
Sorry.
Yeah.
Is okay.
Yeah. Now it's okay.
Okay. And then, of course, we talk about the detection methods. What are the different detection methods by which we can check the virus on the conjunctiva, whether it is RT-PCR, whether it is a viral culture, and whether we have looked at the cytopathologic effects of the virus on the conjunctiva. So what are the evidence available to us? There are a lot of reports published about the conjunctival swabs being positive, and we have also written about why this conjunctival swab can show positive, and that too published in NEJM and JAMA. And we have written about whether it can be a source of contamination. Again, conjunctivitis has been reported in Vigio. So this paper is to highlight about our thermal test strip technique about in 17 COVID-19 patients. None of the patients did demonstrated any SARS-CoV-2 virus, despite all the different techniques, despite all this. So sorry for the poor quality slides. But again, the crux of this slide is that there was none of the patient which was showing any kind of virus on the surface of the conjunctiva. Going into the retinochoroidal vascular changes, Alexander Invernizzi from Italy have reported about the retinal changes or the vascular changes, and they have shown how it can affect in patients with the severe COVID-19. Again, our group also in Singapore have reported about the focal retinochoroidal vascular changes, and these are the focal vascular changes which can happen in the patients with the COVID-19. And likewise, we can also see choroidal vascular changes where there is a change in the choroidal vascularity index and change in the choroidal thickness. So where do we stand as far as the choroid is concerned? Yes, the choroid can be thicker and the CVI or choroidal vascularity index can be reduced in patients with the COVID-19. So to discuss, the subjects were tested positive. However, there was low incidence. Again, point very important is that there is a low incidence of positive PCR in patients with the COVID-19. And again, a lot of hospitalized patients, a lot of patients admitted in ICU were showing positive COVID-19 on the conjunctival secretion. So what are the points for and against SARS-CoV-2 causing a localized ocular disease? So for, it has been detected and reported on the ocular secretions. There have been case reports of keratoconjunctivitis and conjunctivitis, and previous reports of other human coronavirus causing ocular manifestations such as conjunctivitis. So these are in for. What are the points against? It is difficulty in establishing direct causation of the ocular signs symptoms due to virus. Conjunctivitis can be a very, very vague sign, can be a very non-specific sign, can happen in any patients, and ocular findings can be reported by ophthalmologists and who are probably not even present at the scene. So patient with conjunctival swabs, they do not have any other ocular signs or symptoms, which is being surprising again. Again, the theory for can SARS-CoV-2 spread via tears? So for human corneal conjunctiva expresses the ACE2 receptor, which Yolanda spoke about, which may theoretically bind to the SARS-CoV-2 and cause infection. Positive conjunctival swabs in patients with no ocular disease suggest that there will be viral spreading in asymptomatic patient in absence of localized ocular disease. So there is an evidence there. But what are the points against? Unable to determine if SARS-CoV-2 invades ocular tissue directly or spreads from nasopharyngeal secretions. Virus has not yet been cultured from any of the ocular secretion. An ACE2 receptor mainly expressed in posterior tissue, not consistent with reported anterior segment manifestations. So in view of time, I'm not going to go into some of these animal studies, but there are animal studies looking into the associations of coronavirus in the posterior segment of the eye. So to conclude, what are the ocular manifestations of COVID-19? Yes, conjunctivitis can happen. Can SARS-CoV-2 transmit via tears in conjunctival secretions? Yes, there are the reports there, but again, it is still very controversial. And what are the proposed mechanism? We need more research. And of course, we do know about the retinochoroidal macrovascular changes. With that, I'd like to thank IUHG for giving me the opportunity to present on this forum. Thank you very much for your patience.
Thank you very much, Rupesh, for that very precise information, which is very helpful. So continuing with this further, Andrea is going to elaborate on what Rupesh briefly mentioned and what concerns us a lot. Can COVID be transmitted through eye? Over to you, Andrea.
Thank you, Vishali. I would like to thank USG for this nice invitation, particularly my friend Manfred, and the opportunity to share my thought on this topic. Actually, during this topic, I will talk about the infection transmission of SARS-CoV-2 through the eye, but also I would like to cover some aspect on the infection in the cornea, and I will touch some work we've done in ocularology in COVID-19 time. I would like to start with this paper that was published in February 2020, which already has all the points we're talking about today. So in this brief letter, already they say that the transmission of what is SARS-CoV-2, this is the regional name that was used at the beginning, the transmission through the eye was ignored. The respiratory tract is probably not the only transmission route for SARS-CoV-2, and the exposure of unprotected eyes to the virus could cause acute respiratory infection. So already in this brief letter, we have a lot of questions, and for some of them, we don't have the answer yet. So the first two questions are, is the conjunctival potential getaway for SARS-CoV-2? And is the eye as a source of transmission? So regarding the first question, I think there are two nice models that have been used in animals. The first one in monkey, the virus was injected in the conjunctiva and was detected in the conjunctiva on the first day. And after that, in the first week, was detected in the nasal and throat. Specific antibodies were detected in the second week after exposing the conjunctiva to the virus. And after that, the mild interstitial pneumonia was found in this monkey. And this showed that the infection to the conjunctiva is actually possible. The second model used golden hamster. And as you can see in this panel here, they infected the animals either by the intestinal tract, direct contact, ocular inoculation, and intranasal. And even after exposing the animal through the eye, it was possible to have the protein expressed in the lung and have a good inflammatory response with elevated levels of interferon-stimulated genes and chemokines in the lung. And this was actually after inoculation through the eye. In addition to that, they found that there was a systemic inflammation that occurred in distant tissues, even without a very high level of replication of the virus. That means that inflammation can spread out in the entire body. It has been shown, and this is still from the early 2020, that the virus can actually be detected in the conjunctiva and can replicate directly in the conjunctiva. So it can infect actually the eye directly. It has been shown also that the envelope protein and spike protein can be detected from conjunctiva from COVID-19 donors, so from patients. Infecting primary conjunctival epithelial cells, these authors were able to show the spike protein in 24 hours after infection. And they also found the increased expression of antiviral protein and inflammatory response from the conjunctival cells. That means the conjunctival cells can respond to the virus infection. This was probably the only paper that shows that the virus detected from the conjunctiva can actually infect in vitro the virus E6 cells in vitro. This actually was the first Italian patient. She was a Chinese woman traveling from north to the south of Italy in January 2020, and she actually got the conjunctivitis. As you can see, there was the conjunctiva was positive for a long time, and the tears from the conjunctiva were able to replicate, or they showed the replication of the virus in vitro. That means that actually the conjunctiva could be a source of transmission. So in just going through several papers on this topic that Rupesh already mentioned some of them, the SARS-CoV-2 RNA can be present and replicate in the conjunctiva. A patient with conjunctivitis mostly showed detectable viral RNA in tears by RT-PCR. Patient without conjunctivitis did not show viral RNA in ocular specimen in general, but in all cases, the viral load on the ocular surface are kind of low. Therefore, the eye is a potential, even not the classical route of SARS-CoV-2 infection and transmission. There have been some nice studies on the possible transmission of SARS-CoV-2 during surgery from positive patient. And these are simulation using the phaco and detecting the microdroplets from surgery. As you can see, using the fluorescein, the surgeon can be the droplets can reach the surgeon, and somebody else counted actually the numbers of these aerosols particles by light scatter laser photometry. This has been done for other kind of surgery, but there is a theoretical risk that vacuum occlusification surgery might present a contamination and transmission risk for SARS-CoV-2 from COVID-19 positive patient, similar to other type of surgery. I would like to touch some points about SARS-CoV-2 and the cornea. First of all, this is a paper showing that the number of cornea that were procured in 2020 was definitely lower than the year before. And the corneas that were distributed for transplant was dramatically dropped in during 2021, but there was a decrease of about 50% of available cornea for transplantation. But one question is, can the cornea transplant or other tissue can be transplanted without any risk? These are the results from the Veneto Eye Bank, so the bank, a cornea bank from my area. From February 20 to May 2020, they collected more than 1,000 corneas from 588 donors. Of these donors, only 3 were resulted positive for the RT-PCR nasopharyngeal test postmortem. And of these 3 subjects, 6 corneas, 2 corneas were also positive for SARS-CoV-2 RNA. However, when we transfer these RNA to the cells, they were not able to infect the cells, so there was no transmission, and probably was just non-active RNA of the virus. We were also able to study the cornea transcriptome from this cornea and from control, and found actually 3 different clusters in terms of gene expression from negative donors, so those are normal cornea, and from cornea from positive donors that were SARS-CoV-2 negative. And this is the cluster of gene expression from positive donors with cornea that were actually positive for SARS-CoV-2, showing that there are differences in gene expression in these 3 different types of situation. And this is the volcano plots showing that the genes that were mostly overexpressed in positive corneas were related to interferon cascade and to pro-inflammatory cytokines and chemokines. Not only these were overexpressed, but were other genes, the interferon pathways, pro-inflammatory cytokines, and interestingly, some editing enzyme other in apple back were also overexpressed in infected cornea or cornea from infected patients. There were also some genes that were downregulated. Interestingly, some is aquaporin 5 and one of the conjunctival cytokeratins. And this probably is related to the some of the dry eye symptoms that the post-COVID patient might have in the long term. So there is a controversy on the from the literature on the presence or absence of the virus in cornea that can be transplanted. The few papers says that there is absolutely no virus in the cornea. Some others show on the other side that there was a presence in SARS-CoV-2 in patients that had COVID, and they found not only the genomic but also subgenomic RNA in the cornea. In particular, one of them showed that the amount of the RNA was relatively high. So the risk for SARS-CoV-2 infection through the cornea or conjunctival tissue is very low. And SARS-CoV-2 transmission through the ocular tissue might be an unlikely event. Actually, I think has no been reported any infection through transplantation. Maybe just one case of lung transplantation at the beginning of the pandemic. This is from one of these papers. See, the cornea was expression from positive donors, expressing the envelope and the spike protein on the epithelium and in the stroma. However, this nice paper show that the SARS-CoV-2 does not replicate in corneal tissue. On the other side, the herpes virus and Zika virus can replicate in the cornea, and the interferon lambda and interferon lambda receptor 1 play the major role in controlling the infection of the virus in the corneal tissue. We heard that AC2 is expressed in different tissues. It is the gate to enter. So the question is, if AC2 is expressed on the conjunctiva or the cornea, on the cornea ocular surface, can help the infection. This is actually the expression of AC2 in the body, and here the eye is not even considered, only retina, but it's very low expression. We and others have been studying the expression of AC2 in the cornea, the conjunctiva. We used the transcriptomic analysis and found that AC2 was expressed in the cornea, the conjunctiva, actually at low level. On the other side, Tempus 2 was expressed at eye level. We also were looking at different potential receptors and proteases expressed either on the cornea and the conjunctiva. Others have been shown similar results. Even this group showed that the expression of AC2 and Tempus 2 was relatively low. Others have shown that the expression AC2 was and Tempus 2 is more related to the limbus epithelium. This is another paper that even show the expression of AC2 in conjunctival epithelial cells line. And these other papers show the correlation between the expression of AC2 and Tempus 2 to pro-inflammatory signals that might increase the expression of the receptor proteases on epithelial cells. In fact, it's been suggested that at least in the conjunctiva, the expression of AC2 is increased in pathological condition, particularly by the inflammation. We were interested on some paper in another area in allergy that suggested the inflammation type 2 might be protective to the virus infection. And we look at the expression of antivirus genes in vernal-capillary conjunctivitis. VKC is a severe type 2 ocular eosinophilic inflammation that affects mostly children. In our experience, being a referral center for VKC, we had in these 2 years only 2 VKC patients affected by COVID-19 without any ocular symptoms. So we were looking at the calculation association between SARS-CoV-2 and VKC and looking at the gene expression of antiviral genes in patient, not in infected patient, but in VKC in general, and use a transcriptomic approach. Knowing the prevalence of VKC in our area, we calculated the odd ratio of VKC to be associated to COVID-19 and found that this number was 0.88, which was not significant, but towards a kind of protection of VKC regarding the SARS-CoV-2 infection. Looking at the expression of receptors, and these are inflamed conjunctiva. As you can see, AC2 and Tempus 2 and other potential receptor proteases are not overexpressed differently from other report. Only Adam 17 and furin were significantly higher in VKC. And looking for a gene, for gene ontology, for defense response to virus, we found 38 genes overexpressed significantly in VKC compared to control. And between these genes, there were some of interferon-related genes and interferon receptors to be highly expressed in VKC that conclude that local persistent allergic inflammation might be protective for viral infection and similar to what happened in asthma. We also were interested to give some recommendation for treatment, especially because we use immunomodulators in VKC, NTC, or severe allergy patient. And we found that in our group of ocular allergy through Europe, that were not too many concerned about the use of cyclosporine or other immunomodulators in definitely not in after COVID and in patient not at risk, and maybe in patients that are symptomatic, but probably this is not the main problem if somebody is infected. Interestingly, I found this nice paper on the potential effect of protection of cyclosporine to the to SARS-CoV-2 infection. In fact, using this molecular docking interaction, cyclosporine to TM-PERS2 and to another molecule that might be another proteases that could be help the virus to enter into the cells, they found that that might be protective. So in conclusion, the ocular surface has different anti-inflammatory and antimicrobial mechanisms, including anti-SARS-CoV-2 system, low expression IC2, high expression antiviral systems and proteases. SARS-CoV-2 can infect the ocular surface, can replicate in the conjunctiva, but not in the cornea, can diffuse to the respiratory tract and to other organs. The transmission to the virus from the eye to other subject is an unlikely condition, and the type 2 inflammation might be protective for infection, and therapies for, at least for ocular surface disease, should not be stopped in COVID time. And thank you for your attention.
Thank you. Thank you very much, Andrea. And now that we understand the virus, we move on to the clinical aspects. I shall be talking about SARS-CoV-2 induced intraocular inflammation. What does it do to the eye? Actually the relationship between eye and SARS-CoV-2 goes long back. It was 1990. Robbins, for the first time, developed an animal model for experimental retinitis, where he used JHM strain of mouse hepatitis virus. And actually, a few years later, this model was named as experimental coronavirus retinopathy, or ECOR. So we do have MHV viral antigens that are known to affect some very peculiar, not peculiar, some specific layers of the eye, that is inner nuclear layer, photoreceptors, Muller cells, and RPE, where the spontaneous virus clearance happens. However, when we come to the ganglion cell layer, this is the layer where ECOR or MHV virus persisted even after 10 days from inoculation. So let's see how can we extrapolate this information about neurotropism of SARS-CoV-2 virus. I don't have to go over this because you have extensively heard about how these receptors are present within the layers of the retina. And very interestingly, in a recent publication from Dr. Ruben Belfort and his crew, they have performed autopsy of the patients with COVID, and they found that there were virus particles as well as spike protein S and neurocapsid protein. Again, that was detected in almost all the layers of the retina, RPE, and the choroid. Now, what does this virus do? We actually did a study where during the acute phase, we are a multidisciplinary hospital. So we did the screening of the patients who were actually admitted in our intensive care. And we had total of 235 patients. 142, we could do their fundus during the acute phase of the disease. And other 93 we did during the convalescent, which was typically 2 weeks to 17 days after the symptoms were over. Interestingly, when we went to the intensive care unit, we did not find any patient during the acute phase that showed retinal lesions or uveitis that could be attributed exclusively to COVID-19. We found just 5 patients in convalescent phase where we had cotton wool spots, but these 5 patients, that is 5.3% of the total, they did have systemic comorbidities which were present, and those included hypertension, diabetes, iron deficiency, coronary artery, and several other diseases that were associated with the virus infection, or cotton wool spots. Now, this is what the symptoms were like. The findings were kind of COVID microangiopathy like we see in HIV. And on OCT angiography, of course, we could see that there were these areas of significant ischemia, which were seen over a period of time, going to be resolving. Now, very interestingly, maybe it's the persistence of the virus or the particles. These cotton wool spots actually were evolving. They were not static. For example, the top one, the right eye, is baseline. Like we saw this patient 44 days after COVID. The right eye does not have cotton wool spot. However, there is a new cotton wool spot that appeared 2 weeks later. Whereas in the left eye, the cotton wool spot was disappearing, which was expected. What do these cotton wool spots or COVID microangiopathy represent? Honestly, there are multiple theories, but most likely it seems it could be just an immune-mediated response to probably virus particles or to cytotoxic T lymphocytic infiltration in the ganglion cell and plexiform layer. Longitudinally, when we followed these patients, they showed the disappearance of these cotton wool spots with the reperfusion. And we also did MRI because there was a publication that these cotton wool spots could actually indicate an associated neurologic disease. It wasn't so, at least in our series, and we did not find any findings on MRI brain of these patients. When we come to the second manifestation that it produces, and that is being reported more and more commonly, are acute macular neuropathy, as well as the lesions of PAM. Now that we are actually seeing with more and more frequently, now that we are seeing more and more of these patients coming to us. What could be the mechanism? Again, we do not have, a very solid evidence, but neuroretinal inflammation with precapillary arterioles and microvascular occlusion was also observed in ECOG model. So a possibility of intravascular immune complex deposit or thrombotic microangiopathy would always remain. The third category where we see is the COVID-induced photoreceptoritis and neovascularization, and I'm going to show you an example of a patient. This is a 50-year-old patient who came to us. He was in ICU for COVID pneumonitis, and he developed visual disturbances on the 5th day. But he was not in our institute, so we don't have record of fundus findings during that stage. We saw him after 2 weeks when he was allowed to go out. This was his right eye and this was his left eye. Left eye showed some hemorrhage, a gray kind of a vitreous hemorrhage. Whereas right eye on fluorescein angiography shows these hyperfluorescent punctate dots, which were mostly in the mid-periphery. And of course, the OCT had these lesions of AMN, are actually more like PAM in the middle layer. And when we did the OCT through these spots that were seen in the periphery, there was disturbances in the ellipsoid zone. And since there are cells, inflammatory cells that you can see even on, like better on OCT, so we assume that this disruption of ellipsoid was probably the inflammatory response of the photoreceptors to COVID virus. The right eye interestingly showed there was decreased vascularity of deep capillary plexus, but we also had this new vascularization, which was a non-sprouting kind of new vascularization. And when I looked at the literature, this kind of new vascularization, which doesn't have fronts, because being a retina surgeon, I haven't really seen these new vascularization in the active phase. So I realized that this was reported as intussusception new vascularization, which was actually seen following chronic systemic hypoxia and very commonly associated with viral infections, not in the eye, elsewhere in the body. The fourth symptoms are the fourth signs which can happen following COVID, and which we are getting more and more reports, are new inflammation in the RPE choroid, or even reactivation of previously controlled inflammation. Now, to be very honest, with so many of population being affected, whether these inflammation are autoimmune and COVID is simply coexistent, or COVID is inciting some kind of immune response for these inflammation to occur, I'm a clinician, I have no clue, but I'm sure we have the esteemed panel, including now Jim Rosenbaum joining us, who will be able to answer some of these questions for people like me. But I am seeing a spate of these patients, all investigations negative, and only history of COVID is positive. So, they are responding actually very well to local steroids and sometimes even at local anti-TNF, but I don't have the long-term history to show the recurrences. These are two of my patients who were actually very well controlled since last 2 years. And recently they developed inflammation. Again, I do not know whether it is coincidental or it just happened, but we do have a series of patients where the inflammation happened because due to the teleconsult, due to panic, or due to whatever, the immunosuppression was stopped. However, for these two patients, they were on regular treatment. The fifth and the last, which I want to open to the panel, is about delayed inflammation and can COVID induce kind of autoinflammation. Now, this is, I'm showing you an example of a patient who was seen by me in 2018 as a case of central serous chorioretinopathy. And, we knew the fundus was normal before he developed COVID, which is not many a times the case. So then he developed COVID and he was diagnosed as retinitis 1 month following COVID. He was given oral steroids. This is all elsewhere. Patient deteriorated. And he also had some rash in the skin that was diagnosed as pyoderm. This is when I saw the patient. Now, patient had this kind of retinitis, also had this vasculitis. And honestly, I did not know what it was. Like, it is post-COVID, but whether it is COVID causing this or something else, we tried to look for it. There is definitely inflammation, there is retinitis, there is fluid, and the left eye had already undergone atrophy. So probably left eye at some point had retinitis, which had resolved and resulted in the development of scar like this. We did extensive investigations, which are not all reflected. However, his blood culture showed growth of staph hominis. So we thought that probably what we are seeing is a part of staph infection. We treated him on antibiotics. He did respond to antibiotics, but to be very honest with you, I don't know, this is the natural resolution, our response to antibiotics, because the right eye was responding and the left eye was developing some lesions, but finally over a period of 3-4 weeks, he did settle down and both eyes look like this. I haven't seen endophthalmitis patient resulting in these kind of scars and atrophy, and vasculitis, whatever we were seeing resolved. So we did not pay much attention to it, and we did not give him any long-term therapy. Lesions resolved, patient not seeing too well because of foveal atrophy, but otherwise, like, we were happy. Till 8 months later, now patient comes back with acute diffuse vasculitis and decreased vision in the right eye as well as in the left eye. Now, this is not something I can attribute towards staph infection 8 months ago. This is something which is happening ongoing. And at the same time, he also had synovial thickening with effusion at the right hip joint. And our rheumatologist did the tap and all, which did not show anything. So it was diagnosed as a kind of an inflammatory response, and patient received steroids and methotrexate. His synovitis responded, and these are the pictures taken just 2 months after the initiation of methotrexate, which shows a very good response. This is my last slide from a very beautiful article written by Perezio Neri about how the virus can probably activate an aberrant adaptive immune response. If that is the case, are we going to see these patients much after their acute phase is over? And what are the implications in the diagnostics and the management is something I think we can take up during the panel discussion. So to conclude, COVID-induced inflammations are evolving, and so is our understanding. And it's important to characterize clinical, imaging, biological markers, because we honestly do not know what we are dealing with. But if we have to move in the direction of precision medicine, we probably got to pay more attention to some of these things. Thank you very much for your patience hearing. And I think with now we move on to the easily most awaited section of this webinar, that is vaccines, because everybody is getting concerned over it. And it's my pleasure and honor to invite Jim Rosenbaum, who's woken up very early morning for him. Thank you, Jim. And he will be talking to us about uveitis and vaccination strategy, a question that is being asked day in and day out to all of us. Thank you, Jim, and your slides are absolutely fun.
Shally, thank you very much for the opportunity to speak. I'm really honored to have this. I think you have too much confidence in my knowledge of the topic, but I'll do my best. So I'm going to give you a little bit of background on vaccines. I'm going to ask the question, are patients with immune-mediated disease a special case? Show you data on the efficacy of vaccines, the need for a booster, adverse effects from vaccines, which I think Carlos will talk about more, and briefly touch on the topic of monoclonal antibodies. My interest in this is as a rheumatologist, so virtually all of my patients are on immunosuppression. And I've also been working with the Spondylitis Association to try to survey patients with spondyloarthritis to get information about whether they are more susceptible to COVID and whether the therapy influences COVID. Our latest publication is an EPUB in Journal of Rheumatology. We've had 5 similar publications, and at the American College of Rheumatology meeting in 2 weeks, we're going to present some information on vaccine hesitancy and attitudes toward vaccination among patients with spondyloarthropathy. This is not an easy topic, in part because I think the pressure to publish on COVID is such that we are frequently prematurely publishing. The data simply are not always adequate to answer the important questions and the essential questions that need to be asked. In addition, the data are evolving. The virus, of course, is evolving. And patients and physicians want a level of certainty which is simply not warranted right now, I think, by the quality of the data. Furthermore, the recommendations that I'm going to share are based on geographic areas. I'm thrilled that 200 people are participating in this meeting right now, but the answers, if you're in India or in Germany, might be very different from the answers if you're in the United States, and in any given state might be different as well. In the US, most of the vaccinations are mRNA vaccines, either produced by Pfizer-BioNTech or Moderna. We also have a few people, a subset, who are getting viral vector vaccines from Janssen. These are similar to the vaccines made by AstraZeneca and the Sputnik vaccine that's coming out of Russia. There's a very exciting peptide vaccine that's manufactured by Novavax. The preliminary data that they've shared is wonderful, but they are having manufacturing problems. So this is not yet commercially available. It seems to be effective and it seems to be very well tolerated, but again, not available. And inactive viral vaccines are being given in China and India. And then finally, there are countries, Cuba and Iran, among others, that have their own unique vaccines, and I just don't have data to show you about those. I showed you early on the publication that we have in the Journal of Rheumatology about how COVID is affecting the spondylitis population. I reviewed the literature on rheumatic diseases and COVID at the time that we published that study and found 17 publications, and I'm sure that there are more now. But I think the best is arguably from De Silva, that was published in Arthritis and Rheumatology. And this is a great study in the sense that it's a large study. It was based on electronic health records in the United States, and they had nearly 2,400 patients who had systemic rheumatic disease and COVID-19. I obviously would like to be talking specifically about uveitis, but I'm not aware of any publications that have been specifically about uveitis patients. So I hope you'll allow me this extrapolation from rheumatic disease. Besides the 2,400 patients with rheumatic disease, they had over 140,000 patients who were their controls who had known COVID-19 detected in these electronic records. If you had a rheumatic disease, your relative risk for hospitalizations was 1.14. And because this doesn't cross 1 on the confidence interval, it's statistically significant. The relative risk for being in the ICU was 1.32, again, statistically significant. If you, the relative risk to develop renal failure was 1.81, statistically significant. And the relative risk for thromboembolism was also statistically significant if you had a rheumatic disease. However, the major proportion of this relative risk was not due to the rheumatic disease, it was due to the comorbidities, such as lung disease in patients with scleroderma or lupus or even rheumatoid arthritis, renal disease, as we see in vasculitis or systemic lupus, or diabetes, as we often see induced from the prednisone therapy. Vaccines really work. So these data come from the Centers for Disease Control website. Here's the hyperlink. And it's from Los Angeles over 3 months this past summer, May, June, and July. And you can see that the vaccinated or partially vaccinated have far less infection than the unvaccinated in the LA County. Hospitalization is similarly dramatically reduced if you're vaccinated or partially vaccinated over these 3 months in LA County. And here you see how the Delta variant has become predominant, May, June, July. Let's look at the unvaccinated. Minority in May were due to Delta, but by June, it was the predominant. And in July, it was close to being the exclusive strain that was causing disease. And this was true in the fully vaccinated as well. They were not, if they got COVID, it was likely due to the Delta variant. I'm now going to show you a series of articles that appeared in the New England Journal 9 days ago. And again, this is a paper from the Centers for Disease Control. They looked at adults only over age 50. They had over 40,000 hospital admissions and an additional 20,000 plus emergency department visits. The mRNA efficacy for reducing hospitalization was 89%, 90% reduction in intensive care, 91% reduction in emergency department or urgent care visits. Encouragingly, even if you were in a high-risk population, such as being over 85, African American, or Hispanic, the vaccine still was working. And almost all the data come from Moderna or Pfizer vaccines, the mRNA vaccines. We had seen some data earlier on that perhaps the Moderna vaccine was more efficacious, but in this study, the two vaccines were comparable. The adenoviral vector vaccine was less effective, 68% for hospitalization, 73% for emergency room, but it was based on a single dosage. And there are data now emerging that if you boost the adenoviral vaccine, it may be close to comparability to the mRNA vaccines. The American College of Rheumatology has issued guidelines for vaccination among the patients with rheumatic diseases. And guidelines, of course, are a double-edged sword. It's very useful, I think, to many of us to have some suggestions that come from experts, but the experts are limited by the data availability. And if we don't follow those guidelines, there are potential implications in terms of mostly medical legal. Despite that problem, it is useful to have guidelines from experts. And 2 months ago, at least, these experts from the American College of Rheumatology said, if you are taking methotrexate, mycophenolate, tacrolimus, or cyclosporin, you should hold that for at least a week after a 2-dose vaccine, after both the first dose and the second dose. The really bad actor in terms of vaccine efficacy is rituximab. And if you are on rituximab, you want to wait as long as you can after receiving the rituximab, ideally at least 5 months. And if you're going to have another dose of rituximab, give the vaccine 4 weeks at least before retreating with rituximab. All of these recommendations depend on how much you need this immunosuppression. And so there are cases where the disease activity is such that you really need to control the disease, even if it means less efficacy of the vaccine. Ideally, I think as a clinician, you'd like to know whether your patient is immune or not. So we can measure antibodies, but the assays vary. Sometimes we're measuring neutralizing antibodies, but not always. And we're never measuring, at least commercially right now, the T cell response, which might be critical. So patients who have antibodies might be still unprotected, and patients without antibodies might be protected because they have a T cell response, or because even if we can't measure the antibody, they're able to make an amnestic and memory type response. A lot of wonderful data about COVID vaccination is coming out of Israel. And that's because Israel was an early adapter of widespread vaccination, and their health system keeps wonderful records. So again, the New England Journal, 9 days ago, Pfizer vaccine is used exclusively in Israel. Over 1 million individuals studied over age 60. This is about the booster vaccine specifically. 12 days after the booster, the confirmed infection reduction went down by a factor of 11, and the likelihood of a severe illness went down by a factor of nearly 20. When they looked at time points earlier than 12 days, there was benefit, say at 4 to 5 days, but the benefit was greater when they went 12 days and beyond. I think we all know that there are adverse effects from vaccines, and I think Carlos may deal with this far more than I am. The most common would be flu-like symptoms, muscle soreness, but there have been cases of thromboembolism, Guillain-Barré. There have been fatal cases of TTP, thrombotic thrombocytopenic purpura, and there is concern that the underlying disease might flare. A few days ago, I turned to PubMed to see how many cases of uveitis had been reported after vaccines, and I found 14 case reports. There may obviously be many more that are actually occurring. But again, looking at the Israeli group, this is from Victoria Fuhrer, a paper that just came out in the Annals of Rheumatic Diseases, looking at the Pfizer vaccine for how it affected patients with rheumatic diseases. First of all, in terms of efficacy, in this bar graph, here you see the patients who had anti-CD20, which means rituximab, either with or without additional therapy like methotrexate or cyclophosphamide. They had the lowest vaccine response. Vaccine response was also lower for glucocorticoids compared to other forms of immunosuppression like methotrexate, and a drug which we don't use very often in ophthalmology, abatacept, but which is approved to treat rheumatoid arthritis. It blocks antigen presentation. That also reduces the vaccine response. The other drugs had an effect compared to controls, but surprisingly, even very powerful immunosuppressives like the JAK inhibitors still allowed pretty good vaccine efficacy. This Israeli paper also looked at whether the vaccine flared the underlying disease. So here's rheumatoid arthritis, disease activity index, psoriatic arthritis, disease activity index, psoriatic arthritis, or just psoriasis rather, disease activity index, ankylosing spondylitis, disease activity index, and lupus, SLE, disease activity index. Orange means there was no impact, but blue meant actually that the patients improved, and gray meant that the patients worsened. And I think what you can readily see is while there were reports of worsening disease, the reports of worsening disease were actually comparable to the reports of the disease improving. And furthermore, because many patients were holding their medications, we don't know how many of these cases of worsening might be related to the interruption of underlying therapy as opposed to an effect of the vaccine itself. Monoclonal antibodies are an attractive option, especially for individuals who have not been vaccinated. And once again, here's the New England Journal from 9 days ago. These antibodies that were reported on in this paper came from Eli Lilly. Regeneron also has antibodies. These antibodies were given intravenously and within 3 days of PCR confirmation of COVID infection. There was a 1-to-1 randomization of getting monoclonals or placebo. Some of the monoclonals, by the way, can be given subcutaneously. And after a month, 2.1% of those who were treated with monoclonals versus 7% of the controls had to be hospitalized. There were 9 deaths among the 500 or so controls, no deaths in the treated. And now Merck has an oral antiviral, which may well replace the monoclonals because it's obviously easier to administer. It's not yet approved in the United States or elsewhere, to my knowledge, but it may well be the drug that replaces the monoclonal antibody therapy. So what I've shared with you has been data-driven, I hope, but I want to spend just a minute telling you about some humanistic aspects of this vaccine development. These 3 are heroes. They each work for Bion. They were, in the case of this married couple, the founders of Bion. And Uğur was born in Turkey. Aslam's father was a Turkish physician, and she was born in Germany. They met when she was a medical student and he was a resident. When they got married, they invited only people who were working with them in one of their startup companies that they had founded. And as soon as the ceremony was over, they celebrated in the way that a true scientist would celebrate. They went right back to the laboratory. My understanding is that there are a lot of Turkish immigrants in Germany. And like in many countries, there's anti-immigrant feelings. But these 2 Turkish heroes have helped to improve the acceptance of Turkish immigration. Furthermore, the head of Pfizer was born in Greece. So through science, Turkey and Greece actually got greater rapport. But my favorite scientist is Kati Kariko. So she is a vice president at Bion. She was born in Hungary. She and her husband migrated to Pennsylvania, and she got training in science at Temple and then joined the faculty as a PhD at Penn. And this was 20 to 30 years ago. She could not get funded. She was fascinated by mRNA. She wanted to study mRNA, and she could not receive funding. Time and time again, she failed, and she had to sort of hook her star to a senior scientist who could believe in her. One of the things that she did is she realized that putting mRNA into a mouse was highly immunogenic. But if she made the mRNA look like transfer RNA by substituting pseudouridine for uridine, the immunogenicity disappeared. And that's been a key to the development of these mRNA vaccines. All of these people are now billionaires. The value of Bion not too long ago was over $80 billion on paper. And Dr. Terici says that that value is great. It's not distracting. She still bicycles to work, but the money allows her to do more science. And Dr. Kariko has now left Penn for Bion. She has a daughter who won 2 Olympic medals in rowing. So most autoimmune diseases do not markedly affect susceptibility or severity to COVID. They may have some effect, but a lot of that effect really seems to be based on the comorbidities from the disease. Most immunosuppressive drugs also do not markedly affect susceptibility or severity, but a notable exception is rituximab, and we need to be careful with other drugs like methotrexate and calcineurin antagonists. All patients who receive immunosuppression, in my opinion, should be vaccinated, and we should consider modifying their medications with those vaccines. There have been flares of immune-mediated disease, but the risk-benefit ratio still very strongly favors vaccination. So thank you very much, and I look forward to Carlos's talk and the opportunity to have a group discussion.
Thank you very much, Jim. And now we move on to Carlos Perezio from MOVIS, who will be talking about the intraocular effects of these vaccines. And this will be followed by discussion. Over to you, Carlos.
Thank you, Vishali, and thank you to the USG for the invitation. It's always a hard act following Jim. I'll do my best. You know, it's he certainly, all of the presentations so far gave us a very good picture of the problems we've had with this new, well, not a new virus, but a new form of presentation of the virus already known in different forms before, and the fight we've had to create defenses against it, and what is happening as a consequence of this fight as well. So let me just see if I can advance my slides here. Yep. So this is what I'm going to talk about as requested by the organizers, and I have no relevant financial disclosures. So as you already heard, in the search for a way to control the real severe problems related to this infection, we've been looking for vaccines, and in December of 2020, the FDA released this emergency authorization for the Pfizer and Moderna COVID-19 vaccines. And that was followed soon after by the Oxford AstraZeneca vaccine, which was authorized by the EMA. If you look at what's going on, there are 42 candidate vaccines and 141 undergoing preclinical trials. So at the moment, what we have in terms of vaccines, currently we have the messenger RNA vaccines, the Pfizer and Moderna, the protein subunit vaccines, the Novavax, vector vaccines, where the Oxford AstraZeneca is included, and so whole virus vaccines. So several case reports and case series have appeared, and Jim was alluding to this potential data of cases being reported associated with vaccinations. So there are new onset cases of JIA, associated anterior uveitis, bilateral choroiditis, bilateral panuveitis with choroidal thickening, cases of VKH, and from Israel, a series of 21 cases of 19 of anterior uveitis and 2 showing phenotype of mutes. Francesco Picchi published a series as well of patients presenting with episcleritis, anterior scleritis, AMN, PAM, and patients with subretinal fluid. Virgo published 2 patients with AMN and PAM 16 days after receiving the vaccine. So there's a question here in terms of the mechanism. Is this a neuroretinal inflammation? We saw the presentation by Vishali in which was showing that where the virus is really living in the ganglion cells. It's possible that is the mechanism for that, but we know these other manifestations are deep in the retina. Intravascular immune complex deposition, that would fit the thrombotic microangiopathy, which we have seen as other manifestations of the viral infection itself. The case of scleritis and episcleritis after the attenuated virus vaccines, is this an effect on local immune system? We have the option also considering, is there a chance that there is a interaction with the RPE, as the case of subretinal fluid shows, could be a CSR type behavior. And the retinal tropism of the virus itself. So the vaccine may mimic biologic behavior of the virus. The pathogenesis is precisely not known, but of course these are the things that have been discussed. Molecular mimicry, antigen-specific cell and antibody-mediated hypersensitivity, or an inflammatory damage was induced by adjuvants included in the vaccine, which would stimulate the innate immune response. So what we decided to do in view of this small number of cases reported and here and there per series, we decided to try to collect cases, and that was a multinational series collected with the support of the IUSG and the IOIS. And essentially, we were looking for patients who had developed an ocular inflammatory event after receiving the vaccine, and we established that should be within 2 weeks following the vaccination. So we created a control of time for when to report the events. There were 40 international centers. The period of collection of data was a 3-month period during the course of this year. And the following information was collected was the type of vaccine received, the timing of the events, including the date of the vaccination and the date of onset of the uveitis, past ocular history, if they had an inflammatory event, what was the type of the event, what is the local systemic treatment history, and therapeutic management and the outcome. So we collected 70 patients that presented ocular inflammatory events within 14 days following the vaccination. Most of them presented with anterior uveitis, with a few patients also showing posterior uveitis and anterior scleritis. These are the demographics here, and you can see that predominantly in terms of the number of patients, most patients developed anterior uveitis, I mentioned before. There was a history of inflammation before a previous COVID infection was very little, very few patients have that. The vaccines were mostly the Pfizer and the AstraZeneca, comparing to the other vaccines. So it's not going to be surprising that most of the events will be connected to this vaccine. Previous inflammatory events. So if you look at patients who had a number of patients that we saw in terms of total cases that had an event, half of the patients more or less had mentioned an event before, had a documented event before. And again, anterior uveitis predominated. If they were controlled for more than 3 months, majority of the patients were controlled, and you can see very clearly there that there was no patients with ongoing disease, but there were patients receiving topical therapy at the time that this happened, very few, and very few patients also receiving systemic anti-inflammatory therapy. Events were similar to the previous event in the vast majority of the cases. So they reported that what was happening now in association with vaccine resembled the disease they had experienced before. Presentation, most patients developed unilateral disease, and again, anterior uveitis most frequently seen, and all the patients with scleritis had unilateral disease. Visual acuity was unaffected in the majority of patients, or if affected, was with a reduction of vision with less than 3 or equal to 3 lines. Very few patients had more significant involvement of vision, none in the scleritis group. In terms of management, the patients received mostly topical therapy, reflecting the fact that there were mostly anterior uveitis cases that were being reported. Systemic therapy was also used, probably reflecting the other manifestations that we saw, and there were also use of other forms of treatment, including antivirals, non-steroidals, and antibiotics. The visual outcomes I mentioned before, visual unaffected in the vast majority of the patients. The reduction in vision was not significant also, but the few patients, I'll mention to you which ones, had a reduction of vision of more than 3 lines. Complications, again, we talk about the vision, I already mentioned that to you. Visual involvement of intraocular pressure was not significant. There were some corneal lesions in one of the patients was a reason for the reduction in vision, and cystoid macular edema was also reported. And was a patient that developed macular scarring, which is the reason for visual, one of the patients mentioned as having lost vision more than 3 lines. Was there a difference between first and the second dose? So the time after the first dose, you can see here a total was 6 days, between 1 and 14 days, and 18 patients received the second dose, and 33% of those patients had a recurrence after the second dose. So this would be, when we talk about how we really feel that a vaccine is connected to the event, because this is a discussion that I had with Mark DeSmet, who's also on this call here today, that, considering the number of vaccines that we have seen in the world, it is sometimes questionable if an event reported now is truly related to the vaccination or could represent just a isolated event. But when you have a recurrence after a second dose, that is when you really feel more inclined to believe that there is a true association with that event. And the event, again, the time after the second event was very similar to what was reported with the first one. Anterior uveitis, we can see here 41 cases after vaccination, and a lot of patients were patients known to have HLA-B27 anterior uveitis. Interestingly, one of them was using secukinumab, and one was using infliximab and methotrexate. You had some patients with idiopathic anterior uveitis and other forms, including viral disease and JIA, mentioned also in another report before. So there were several different types of events, predominantly the HLA-27, and all the events after vaccination to these patients were similar to what they had experienced before. Visual loss, we had 2 patients, and one had the innumerable corneal lesions I mentioned before, and one patient had macular edema. Posterior disease, had 9 patients had posterior uveitis after vaccination. There were cases of recurrence of toxoplasmic retinitis, cases of new onset of toxoplasmic retinitis. There were patients reported multifocal choroiditis, and cases of choroiditis and retinal vasculitis. So one patient with ocular toxoplasmosis and one with the occlusive retinal vasculitis had persistent visual loss on the last follow-up due to macular involvement. Scleritis was seen in 7 patients, and there was a history of idiopathic anterior scleritis. There was a history also of idiopathic posterior scleritis using methotrexate and prednisolone, and there were 2 cases of new anterior scleritis. So all the events were similar to their previous one, and none of these patients suffered any visual loss. These are other events that were reported, including optic neuritis, and the discussion we had last week in the USICOM meeting, in the India meeting. One of the colleagues said that she had been aware of her patients developing optic neuritis and having severe disease. So in this group here, the optic neuritis responded to treatment. There's no report of this patient losing vision, very few, only 2 cases reported. And we also had experience with patients that we saw in our institution who responded to treatment. Other events, the history of intermediate uveitis, idiopathic uveitis, optic neuritis, episcleritis, CMV, anterior uveitis. So there are other events that were reported. So the events of the after vaccination was similar to the previous ones, except for the patient, a history of CMV anterior uveitis who presented at the time of these presentations related to vaccination with intermediate uveitis. One patient had persistent visual loss due to macular edema in intermediate uveitis. The patients were managed, a lot of them, as I mentioned before, with topical therapy. There were patients receiving systemic steroids, 5 were combined with topical therapy and 4 with antibiotics, and no treatment was observed in 14% of the cases. And there were the other reports of topical steroids and non-steroidals, IOP lowering medications. And so these were all reports, just showing to you how the clinicians were involved in the care of these patients reacted to the events that they were witnessing following vaccination. This is the issue about how do you establish the real relationship between an event and what you see. And these would be the 9 points that would be considered. So the strength of the association, consistency, specificity, temporality, biological gradient, plausibility, coherence, experiment, and analogy. So from what we see here, temporality was demonstrated because we created these 2 weeks and the events were occurring within that period of time. Exactly reproducible or not, we already told you that there were 33.3%, 6 cases within the patients who received the second vaccine who presented similar events following the second dose. And different observations in different regions from different people would support the consistency between the exposure and the outcome. How plausible it is? Well, the causality is plausible as some biological mechanisms have already been proposed, as I mentioned to you before. In analogy, the events are analogous to those reported following other vaccines as well. So this creates a reasonable belief that what we see is a true association. In conclusion, most of the inflammatory episodes that we have seen were not severe. Most of them were anterior uveitis. Most of them were unilateral cases. Patients were mainly managed with topical therapy or observation. Unaffected visual acuity was predominantly what we saw. And there were very few patients who ended up really losing vision as a consequence of different problems related to these events. I thought some points I'd like probably to raise for discussion is something that came as following the meeting we had last week, and considering the risk of a second event coming when you have a revaccination, would it be important to check antibody levels for those who presented with a reaction and then potentially delay that second vaccine? Would be the way that would work. And that's something that I'm sure we can bring up in our discussion following my conclusion here. I'd like to acknowledge here, these are all the members of this work, all the centers who contributed data for this study. So I'd like to thank all of them. And the paper now is being submitted to British Journal of Ophthalmology, and we are expecting the comments back and hopefully the publication in the near future. Thank you all.
Thank you. Thank you, Carlos, for a very, very interesting talk. And now we move towards discussion, and over to you, Manfred. Manfred, you are muted. We can't hear you.
No, I'm not muted. Sorry.
Yes, now, but your voice is...
That's better.
Yeah,
Can you hear me now?
Yeah. Now it's
okay. So thanks a lot. So let's try to a little bit, some kind of controversy probably. And I have a few questions to, I think, for all of these, more or less all of these presentations. Number one is, what do you think about new mutations in the moment? They are still very important for Europe at this moment when we have such a lot of up to 80% of vaccinations. Yolanda, any idea about this? Is there real danger in this moment?
We should also always be vigilant, but I think the vaccination rates are pretty high, at least in Western Europe. But in Eastern Europe, there is really a delay in vaccination. So there we still have a lot of viral replication and spread. So there may still be variants of concern coming from that direction.
Would you agree that in Africa, people are less in danger compared to European Americans? Any idea about this one? I think there are really much lower numbers in Central Africa and in all of these countries. I think the one which have the highest one besides Tunisia is South Africa, but in between the other ones, there's not so much. Are there any factors known about this, protection factors?
Well, the thing is, as long as we still have a very large population that has never encountered the virus. So it's always a race against the clock. If you get vaccinated before you see the virus, the chances you become ill or even die are significantly reduced. So even though the number of infections are low, if we don't get vaccinations there, people are still will become infected in time, and that may be next week, but it may be in a year or 2 years. So
we also have a very young population in Africa, so that should population being younger, statistics being not what they are elsewhere, may also influence the appearance of a lesser rate of transmission. I don't know if there's good data to suggest that it would be very different in Africa.
But especially young people get infected in Europe like hell. In Germany, they have, I think, 70% of new infections are young people. So from there, they should also infect the old ones.
We're testing a lot in Europe. Not all of those 70% are ending up in hospitals. So a lot of it is based essentially on traceability. And that probably is less, I think, in the African continent. The data I saw from Africa is in those countries where there is tracing, the numbers are in fact going up. There may be a delay, but they're not that low. I don't have the exact statistics, but they're not really much lower than what you would see in other countries as a pickup occurs.
But comparing to Brazil, I think it's still quite low. So big difference with India.
Trying to make a comment.
Well, I'm far from an expert, but I would think that capturing cases, tracing, as Mark said, is really critical. And it's hard to know how much we can trust the data out of Africa. On the other hand, it's very clear that genetics affect viral illnesses and other infections. And so it's possible that they won the genetics lottery for this one, but I think that remains to be seen.
Mhm.
I had a question maybe to Yolanda and maybe also Jim, if you would like to contribute. Is the virus has been around for a while. We've had some new variants that have come along. And if we think about a virus trying to infect and it's creating variants continually, would the most virulent ones already have appeared? And as Manfred was bringing up, we may still have some new ones. If we get new variants, do we have to be as worried about those, or should we expect them to be a little bit less virulent and more in line with what we've, with a less infectivity, let's say? It's an interesting question. I don't know if we can have an answer, if we should even suggest that, but there are only so many variants that are possible in some ways.
That's true. And yeah, this is a very interesting question and very difficult to answer because I can't look into the future. But yeah, I think you may have. Yeah, go ahead, Jim.
I hope that the opposite will happen. That what's going to predominate are the viruses that are most infectious. Whether they'll be the most virulent or not, that's difficult to say. But evolution. The virus is trying, not consciously trying, but it's trying to do all it can to wreak more havoc.
Keep going.
But Jim, we've been around since the AIDS period, and in the AIDS period, it killed a lot in the beginning. It's now maybe transmitting a bit less, and it is somewhat less virulent than it used to be, despite all the medications we're using right now. So, there's an evolution on the virus side also, let's put it that way. And I think, maybe we have to look at that too.
I think
we have two participants from Africa who want to say something. One is Jenny, who is from South Africa. She says that testing rates are very low in most of the Africa, so the numbers are not accurate. And the low vaccination rate is going to make a high risk for developing variants, like what all of you are saying. And then there is Josephine, who says that my patients with relatives in Central Africa tell me things are very bad there. It's just not being reported. So just to carry on.
I'm going to make one comment here on the back of what Jim and Mark was saying. I think in terms of evolution, Jim, if a virus is killing people or creating severe disease, that virus is more likely to disappear, right? Because it's going to be isolated more readily. Those patients who are very ill become isolated, or either they die or they survive, but they're not contacting more people. While the virus, which is milder in manifestation, is more likely to go around without being detected and spread more easily to the population. So it may be that the issue of the variants that will be around is that the variants that will cause less severe disease will be probably less detected and easily, and the contact's going to spread more rapidly rather than the ones that are really making people go to hospital and be admitted or die as a consequence of it.
But Delta was the opposite, isn't it?
What is that, sorry?
Delta was the opposite. It was more dangerous, definitely. We
No,
all
it's dangerous
expected
because it
lighter
kills
versions, but
Yeah,
Delta is,
Yeah, if you kill people more, it's more dangerous. But in terms of
Sure.
spread, I wonder if in terms of what Mark was suggesting, in terms of long-term, what we may see, would that be a factor that might affect what we tend to see of progression of these strains that are coming, these new variants coming?
Let me ask you one more question.
100 years ago, we had no vaccines and we had a flu epidemic, and it did eventually disappear. This epidemic has had more waves, I think, than prior epidemics. And clearly there are things we don't understand about the coronavirus. So an optimistic approach is, yeah, it's going to go away. But
I don't think so.
Yeah.
I think the coronavirus is here to stay. If we look at the other coronavirus, they tend to reappear. Well, after 6 months, we don't become fully immune to this virus. Our immunity wanes. So I think in the end we will end up with another respiratory coronavirus, which every now and then pops up in the community, and in some people will cause severe disease, and most will have a snotty nose and be fine within a couple of days. So in that sense, Mark, you're right that eventually we will end up with less virulent types, but there will be around. But we're still, and this is a situation where the entire population, with the exception of the young children, has encountered the virus and has mounted some kind of immunity against those viruses. We still now have populations which are completely naive, and these are still vulnerable. So, and there you may have a situation where you have massive replication of the virus and perhaps still some quite virulent
so in other words, we can hope,
from that
but our guard,
still.
yeah, so we can be hopeful, but our guard has to stay up at the present time.
We have to stay
time
vigilant.
To
yeah,
yes.
exactly.
Can I quickly
and vaccinate?
Sorry.
And vaccinate, yes.
Can I quickly change the topic a little bit? We still have two topics I think we should mention. One is, I think, very important for all of practitioners. When would you expect a COVID-19 induced situation in the eye? So every red eye, in addition to COVID positivity, probably, yes. What about intraocular inflammation? Where do you see a combination? It's a time problem.
Well, I tried to look at maybe somebody else wants to answer, but I tried to look at that based on the ECOR. I went through all the papers that John Hooks essentially wrote. And it would seem that the virus, at least in the mouse, lasts for about 10 days. The RNA can be detected even nearly a year later, although apparently protein is not expressed. So the virus is there and may be replicating at a very low rate in the retina as well as the RPE. And the retinal degeneration that is seen is both retina and RPE seems to start somewhere about 10 to 14 days later and is ongoing for a year. So presumably we should still expect to see people appear with some manifestations later. And I think a lot of the immune response, if anything from the vaccination as well as the patients described so far, is often starting about 14 days to 21 days after the original disease has started to quieten down. It's usually about a month after the first symptoms. So it's somewhere 2 weeks onwards. Would you agree, Vishali, from your experience, because you've seen quite a few cases in India?
Even after vaccination, these, something which is not usual, it's multifocal choroiditis, not the usual. We recently saw, which I shared with Carlos, patient with mutes, but it's not the usual mutes that we see atypical, but they are not all typically within 2 weeks. Even after vaccination, we are seeing it 4 weeks, 6 weeks, 8 weeks, and all the etiologies are negative. And somehow they are not typical. You just look at the patient and you ask, has the patient got COVID or COVID vaccination in the past? But there is something different about these patients, which I'm not yet able to put my finger on, but I'm sure we would.
The other thing I find fascinating also is if you look, there are cases of what looks like ampy or serpiginous that are correlated with the fact the person has been vaccinated or has had, the infection. And we've never been able to pin any kind of disease on, serpiginous. And we've always thought that there was something there that might be of viral origin. So maybe, these coronaviruses will help us understand better some of these deep diseases that are affecting, the choriocapillaris, the RPE, and the choroid.
And Annabella has just written a comment that they are seeing a lot of patients of leakage in multiple centers in Japan following vaccination. So what is your take on that? Anybody from the panel?
Jim.
Well, so, we talked about hope that the virus is going to go away. The real hope is that we're going to be smarter from what we're learning about this. The HIV epidemic was a tragedy, but we learned a lot about T cells. We learned a lot about viruses. And I do think that there are analogies. The Steve Ye and his colleagues, what they've taught us about Ebola and the eye immune privilege for viruses and the balance between the immune response and what the virus does, which we think is HSV in the cornea also. And ARN cases are this difficult tightrope balance between what's the virus doing and what's our immune response doing. Immune recovery, uveitis in HIV patients. Another great example that the antigen just lingering around can do a lot of damage. So maybe we will much better understand VKH thanks to the virus. That's our gratitude to the virus.
Do you think it's just inciting the inflammations that are otherwise common in that particular geographic region? What I mean is serpiginous for us post-COVID, VKH for Japan, and could it be explained?
Well, my own feeling is that a lot of these immune-mediated diseases are a perfect storm, that it's not a single factor, it's not a virus all by itself. There's wonderful experimental work on celiac disease where you take the mouse, you give the mouse the HLA type that predisposes to celiac, nothing happens. You give the mouse a diet with gluten and the human HLA, nothing happens. And then you infect the mouse with a virus that by itself doesn't cause any manifestations of disease, but it infects the gut, and voila, you start making antibodies to gluten. So yes, the answer is, I think that there's a perfect storm, that the reason why we don't fully understand VKH or serpiginous or any of these diseases is because it is so multifactorial, and we're going to get clues from coronavirus infection.
Jim, the one point is just wanted to make very quickly, Vishali and Jim. Normally you don't see viruses causing primary choroidal pathology. It's unusual. You see that even ARN with a severe retinitis, you look at the choroid, is usually not involved. So what I think the point you're making is very true, that what we're seeing is this stimulation of the immune response, which is being induced by the virus, is probably interfering with other problems that would have happened and have been now induced by that immune status that has been modified, rather than being primarily an effect of the virus specifically in there. So these VKH patients will be, the immune system is doing something that is now allowing VKH to show itself, but it doesn't mean the virus is in the choroid causing the VKH to occur. That was my way of thinking of it.
You know, one question which is coming repeatedly for Carlos and Jim is, what if uveitis happens after first dose of vaccine? What do you do about the timing of the second? And there is also a question about third dose of vaccine.
Yeah.
I wish we had more data about switching horses in the middle of the stream. That is, if it occurred after an adenoviral vector vaccine, should we switch to a protein vaccine? It would be nice when Novavax is available, as an example. But I would, there is some immunity from a single vaccine, and you just have to weigh the severity of the eye manifestation. And I would seriously consider switching the type of vaccine for a booster dose.
And
Just want to make one comment.
I'm giving, no, go ahead, Mr. Pavishek.
There's just a very quick thing. The data that I showed, I think what the point I failed to make at the very end is that the data supports the idea that even though events can happen, they're usually mild and patients should carry on with the vaccine. It is exactly the question, should I have my vaccine because of this? The risk is small, and if it happens, it's mild. You don't tend to lose vision. So yes, by all means, the patient should go ahead and be vaccinated. The issue of the second vaccine is something we can see that there are patients getting inflammation following the second one, and we can see if the first event was very severe, then you might say maybe the second vaccine, you need to think about it. That's what we're discussing, measuring antibodies, delaying the second vaccine. We have to find different strategies for that. But by far, go ahead and have your second dose. Don't stop there because it's unlikely to be serious.
Carlos,
just a point to add.
Just
a point to add there is we can give them prophylactic steroids, which I'm giving for my patient who had after the first dose a mild reaction or a severe reaction, and before they go for their second dose, I am prophylactically starting the either topical steroids for anterior uveitis or systemic steroid, and which has significantly blunted the effect and probably aborted the attack which could have happened after the second dose of the vaccine. We had at least 3 or 4 patients of the weak age disease. One of the patients presented with the same day, but a lot of them were treated, and I agree with Mr. Perezio that most of them respond very well to the treatment. So I think that's
Carlos,
a good news.
One question. Did you check the antibody response in the patients who developed uveitis, a recurrence of their uveitis?
No, it wasn't checked. This study was collecting the cases that were reported.
We did a few, and they all had good antibody responses.
This is what I raised
despite immunosuppression or something like that.
Yeah. That's the question. Part of question, Linda Lim from Australia is asking that if post-COVID there is a severe uveitis, would you give vaccination or not? So would you measure antibody titers, and would that help you decide?
But that's the point about the between first and second vaccine and potentially what you're saying, the real COVID and the vaccination. So if some the case, clearly the different types of manifestations in the eye, which Mark presented last week and Jim alluded to today, of infection, things that PAM or AMN, or things that are not necessarily inflammatory in the sense of a uveitis, but they are related to inflammation at a more localized level rather than the very inflammatory eyes that we see. Your question is, would exposure to the vaccine create an issue that I will potentially lose vision as a consequence of that? And I don't think we know the answer to that. I don't think we can answer that question. I think if you had a serious event related to an infection, your concern is what will happen if my immune system is tickled again with this virus, and then what will happen to me? I think is a great question that I don't think we can answer. But from the point of view of the vaccinations and the question that Manfred was asking, if you have a high antibody response, you may very well consider that vaccine could be delayed, because the whole intention of the vaccine is create a good immune response. If you already have a very strong immune response, there's a point in delaying the vaccination and then allowing potentially preventing some of these cases.
And
I think what would be nice is to know. Go ahead.
I was just going to make the point that it's a moving target, as the monoclonal antibodies, Merck's new drug, Novavax's vaccine, all of those things are going to change the equation, and it's shared decision making. We could do this. This would be my fear. How much are you afraid, and how likely is it that you're going to get COVID? And let's make a decision together.
And
I think, it's good to have antibodies. It's good to have antibodies. The other is, of course, we need to know more about the cellular immunity, which is really key for viruses. And then, as a follow-up maybe on your point, Jim, is that now in many parts of the world, at least in Europe and the US, more and more people are vaccinated. Putting a delay of 2 to 3 months between the first and second dose might not be such a bad idea. You reduce the amount of antigen that is there, and maybe you reduce the risk of having an immune response, and you don't have to fear as much getting ill after your first dose as we did just a year ago, for example.
Yeah.
So you have to look at what you said in the beginning is very true. It depends on where you live.
Yeah. Is the Moderna vaccine superior to the Pfizer Bion vaccine, or was it that one was 3 weeks apart and the other was 4 weeks apart? I mean,
exactly.
And Annabelle also wants to inform us that post-vaccination weak age patients are actually not very severe, are no worse than the usual weak age, and responding well to treatment for.
That's also in our series, yes. So I think if there is not very important questions anymore available. We are over time already, 4 minutes after. Any more questions which are very urgent?
The last question, would systemic steroids blunt the effect of vaccine?
Dr. Vishal, if I can take that, because see, the uveitis patients, the rheumatology patients are on biologics, are on systemic steroids, and we do give them the vaccines. And that's been recommended. And as Professor Rosenbaum also said, that there is nothing which we need to stop them from not taking vaccines or for the fear of it. And again, it brings to the same question also, if the patients are on systemic steroid, are they at increased risk of getting COVID infection? And this was the question we did ask when the COVID infection came up and then there was no vaccines. So I think probably no, my answer will be, but Professor Rosenbaum, if he wants to say some other points related to this thing, so I don't agree to this, that systemic steroid will blunt the effect of COVID vaccine.
I think it depends a lot on dosage and duration, and we don't have adequate data.
One thing to mention on this, just for curiosity, it's not curiosity, but it's what's happening. In the UK now, what you're doing, if you had you were immunosuppressive therapy at the time you took your first doses of the vaccine, they're offering you now a third dose, not a boost. They're offering you a third dose, and the boost is only for the people who are not immunosuppressed before. So I took a boost, but immunosuppressed patients are offered the third dose of the vaccine as an attempt to increase their response.
Okay. So with this one, I would like to thank all participants for a wonderful webinar. I enjoyed it a lot. I hope that our people who followed, I think it was up to 200 people, also enjoyed this meeting. Thanks so much. And we meet again, I think, beginning of November for the second part of the whole series, which will be HIV. Thanks so much. For the people which are on the board, can you also have a short look for the question and answers so that we can finally solve these two?
Yeah, we are doing that. Thank you everyone for your time and efforts.
Thanks, everyone. Jim, you can go back to sleep now.
Yeah.
Thanks. Thanks for
Nice seeing you.
Okay.
Take care.
Nice seeing you. Yeah.
Take care.
Thank you. Bye-bye.
There is probably one
Bye-bye.
for you about retinal hemorrhages post-vaccine. Rest everybody is done. Thank you very much.
Bye-bye.
Thanks, Yolanda. See you later. Bye.
Bye.
Bye-bye.